Hey guys, so today we're going to be talking about photosynthesis and cellular respiration, and we're going to talk about how they're related, why they're important, and what you need to specifically know about them. So first we're going to talk about photosynthetic organisms. Those are algae, plants, and cyanobacteria. Cyano just means blue, so they're blue bacteria. And all of these can transform solar energy into chemical energy of carbohydrates. If we didn't have solar energy, if we didn't have the sun, these plants and other organisms wouldn't be able to produce their carbohydrates and therefore wouldn't be able to sustain life on Earth. We would have less oxygen because one of the byproducts of photosynthesis is oxygen. And also we wouldn't have anything to eat because whether you're a vegetarian or a meatitarian, you depend on fruits and vegetables and grasses and plants for your food. So photosynthesis captures solar energy and turns it into chemical energy, and it stores it as a carbohydrate, whether that be a simple sugar like glucose or fructose, or if it's a complex sugar like starch or cellulose. And photosynthesizers, so those photosynthetic organisms mentioned above, are producing food energy for not only themselves, but also for heterotrophs. And heterotroph just means that they eat something outside of their body. They can't make their own food. So photosynthesis takes place in the green portions of plants. So a lot of times we think of like leaves on trees or leaves on flowering plants. And the chloroplasts absorb all of the other colors of light and reflect green. So that's why they appear green. The raw materials for photosynthesis are carbon dioxide and water. The roots of a plant absorb water, and that moves up their vascular tissue. So they kind of have veins and arteries like you or I have, but instead of moving blood, they move water. And then to get carbon dioxide, they have small openings called stomata. And that allows carbon dioxide to come into the plant cells and diffuse into the chloroplasts. In the stroma, which is the gel-like fluid that's around the thylakoids in the chloroplast, the CO2 gets combined with water to form C6H12O6, which is sugar. And the energy that's needed for this, instead of coming from ATP, like an animal uses energy from ATP, it comes from light. So they absorb that solar energy. And like I said, they absorb those wavelengths that aren't green, and they energize electrons. So they break bonds to release electrons to reduce CO2 to a carbohydrate. This is a picture of a cross-section of a leaf. You can see that there's something called a leaf vein. So that's where that water is going to come up and interact with those cells. You can also see the stoma, which are those little holes. Lots of plants can actually close those off so that they don't lose so much water vapor. And also it's where O2 is released. And you can also see the chloroplasts and the thylakoids that are inside those chloroplasts where the photosynthesis actually takes place. So the process of photosynthesis is broken up into basically two parts. There's the light reactions that have to have light to happen. We can simulate solar energy by using heat lamps as we do in greenhouses or grow houses. But these light reactions can only occur in the presence of light. And they do capture energy, and the chlorophyll absorbs that solar energy, and that energizes those electrons. And when those electrons move down an electron transport chain, which kind of looks like, kind of functions like a paddle wheel, it pumps hydrogen ions into the thylakoids, which creates a concentration gradient. So if you remember, from the cell membrane when there's a high concentration on one side and a low concentration on the other. When they open the floodgates, as it were, and it pushes through, then it can be used to paddle a protein around that generates ATP. Then there's the Calvin cycle reactions, and they're named after the guy who discovered them, and they take place in the stroma, and that's where CO2 is reduced to the carbohydrate. And the ATP that we created in those light reactions are what's used to power the Calvin cycle reactions. So you can see that the solar energy comes in to the chloroplast, goes into those thylakoids where the light reactions happen, and then that energy is used to power that Calvin cycle reaction. So there's different pigments that are used to power these light reactions. Not only is there chlorophyll, which is the one that we know the most about, but there's also carotenoids. And they absorb certain wavelengths of light, and some are absorbed and some are transmitted. The carotenoids are what you see in the fall and winter that are orange or red. And then, of course, chlorophyll is the one that appears green. This graph shows the visible amount of light you can see is a very tiny portion. And even that is broken down into all the way from purple to red, with purple being the highest energy and red being the lowest energy. And you can see that chlorophyll A absorbs a lot of that purple blue spectrum. The carotenoids also absorb a lot more of that purple blue, almost into green section. And then chlorophyll B gets a lot of that blue. And then it also picks up some of the orangey reds on this side, but it reflects all of this greeny yellow here in the middle. So this is the entire wavelength spectrum here, and only a small amount of that wavelength actually gets to us. So only about 42% of the solar radiation that hits Earth ever reaches the surface of Earth, because it's either filtered out by the ozone layer or by water vapor and carbon dioxide. So the light reactions consist of two alternate electron pathways. There's the non-cyclic and the cyclic. They capture light energy with the photosystems. There's photosystem 1 and photosystem 2. That doesn't it doesn't refer to the order in which they happen. It's just the order in which they were discovered. And so a photosystem basically consists of that pigment complex and an electron acceptor within that thylakoid membrane. So either chlorophyll A or chlorophyll B or one of the carotenoids, and then either NADP or NAB or FAD that is willing to take those electrons on, so that they can then be broken apart later to create that hydrogen gradient. Both pathways produce ATP, and the non-cyclic pathway also produces NADPH. So in this non-cyclic, it takes place in the thylakoid membrane. Photosystem 2 captures the light energy, and that causes an electron to be ejected from the reaction center, which is chlorophyll A, and it travels down that electron transport chain, basically like going down the stairs, until it gets to PS1. And then it's replaced with an electron from water, which is split up from into O2 and H positive. And this creates a concentration gradient of hydrogen ions to accumulate in that chamber. And then that gradient is used to produce that ATP. So when it goes out of those thylakoid chambers, when it goes across its concentration gradient, then it starts to cycle those ATP synthases, which are what those complexes that cause ATP to be formed, and it makes them basically move so that the ATP can be produced. Photosystem 1 captures light energy also. It also ejects an electron, but the electron is transferred permanently to the NADP, which creates an NADPH. So you can see here a visual representation of that non-cyclical electron pathway. And those products that it creates, that ATP and that NADPH, goes on to power the Calvin cycle. So here's another visual representation in a little more depth. It goes down this electron transport chain. So this is that stair step I was talking about until it gets to photosystem 1, and then it goes up that electron chain in photosystem 1 until it creates that NADP, okay? So the electron transport chain consists of a cytochrome complex, and it carries those electrons between photosystem 2 and photosystem 1. Photosystem 2 has to have a constant influx of those electrons because they won't recycle, but photosystem 1 can recycle its electrons through that NADPH. The photosystem 2 has to have a constant influx of H2O to get those new electrons back. And then, of course, we talked about the ATP synthase complex, where it has that channel flow for those hydrogen ions. And when it drives that through, it drives that to join ADP and phosphorus to make ATP. So this is that ATP synthase right here. So those, you see there's a high hydrogen complex concentration gradient on this side, and it goes through, which makes this ATP synthase work, and that ADP joins with another phosphate to create ATP. And that's called chemiosmosis. So the thylakoid space acts as a reservoir for those hydrogen ions, and each time water's oxidized, 2 H pluses remain. So 2 of those hydrogen ions remain in that thylakoid space. And the energy that is yielded from that electron transport chain pumps those hydrogens across that membrane so that it will continue to have a higher concentration on the inside than it does on the outside, so it can continue to use that concentration gradient to create ATP. One of the impacts that we need to focus on is destruction of the tropical rainforest and climate change. So when we start to destroy plants, we're destroying ourselves, basically. So tropical rainforests can exist anywhere where the temperatures are above 26 degrees centigrade and where rainfall is between 100 and 200 centimeters regularly. Most of the plants in the tropical rainforest are woody. There's lots of vines, and there's very little undergrowth on the bottom. So you're not going to have a lot of bushes. But it contributes greatly to the CO2 uptake. So taking in all of the carbon emissions from our cars, from our electrical plants, from the immense population that we have. There's almost 7 billion people in the world, and that's a lot of CO2 when you think about it. And when we destroy those rainforests, it reduces them, and it reduces that CO2 uptake. So you can see from this slide that development in those countries where the tropical rainforest has reduced them by 8%. And the deforestation accounts for 20 to 30% of atmospheric CO2. So it's not only is it decreasing the amount of CO2 it can absorb, but it's increasing the amount of CO2 because we're burning them or we're using energy to transform them into paper and all kinds of things. And it also increases temperatures because that CO2 acts as a blanket and it keeps that warm air in. Now, it's not going to keep that warm air in one area, so you get extreme temperature changes in certain areas. So we really need to focus on how we can prevent the tropical rainforest from being destroyed, or some way to combat that when we do develop it. So you can see that the global temperature change has increased from 1860 all the way up to 2020. We've increased 3 degrees. Now that may not seem like a lot, but when you're talking about homeostasis and trying to keep things in balance, 3 degrees can be a lot. So we talked about how when we destroy the forest, we destroy things that fix carbon dioxide. That means that they take in carbon dioxide. So there's a cyclical set of reactions that allows plants to absorb carbon dioxide. And it's the first step of the Calvin cycle. And during that reaction, 3 molecules of carbon dioxide from the atmosphere are attached to 3 molecules of what we call RUBP, which is a sugar, a ribulose 1,5 bisphosphate. So there are, this is known as C3 photosynthesis, and we'll also see something called C4 later. And it has 3 stages. So it involves carbon dioxide fixation, carbon dioxide reduction, and RUBP regeneration. So like I said, that CO2 is attached to that 5-carbon sugar, and it results in a 6-carbon molecule. And then that splits into 2 3-carbon molecules. The reaction's accelerated by an enzyme, which we usually call rubisco. And then CO2 is now fixed because it's part of a carbohydrate. So that 1 carbon from that carbon dioxide is added to that 5-carbon sugar, and that's fixed into those carbon molecules. So so O2 is then released. So if you look at this visual, you can see where that CO2 comes in, and it's fixed into these 3 CO2 here. And then it goes through reduction and the regeneration of the RUBP. Because if you don't have that RUBP, then you don't have anything for that carbon to attach to. So the 3PG, so that 3-phosphoglycerate, or that 3,6-carbon sugar, is reduced to something called 1,3-biphosphoglycerate, which is BPG on this slide. And then it's reduced into glyceraldehyde 3-phosphate. And basically what that means is it just keeps getting reduced until it's something that the plant can use to make either glucose or another type of carbohydrate. It does take some NADPH and some ATP, but those are generated in those light reactions, and so that's helpful to drive that carbon dioxide fixation. So you can see here that it takes that ATP to make it from 3PG to BPG, and then it takes that NADPH to change it from BPG to G3P. And I know that's a lot of numbers and letters coming to you rather quickly, but such is the nature of photosynthesis. So then we have that regeneration of the RUBP. Because it has to be used for that carbon fixation, it has to be replaced. And every 3 turns of the Calvin cycle, we use 5 G3Ps to remake 3 RUBPs. So we use 5 3-carbons to make 3 5-carbons. So you're not losing any carbon there. It's still 15 carbons altogether is what that bottom equation is trying to show you. And it takes 3 molecules of ATP to do this. So why is the Calvin cycle important? because we can change those G3Ps into pretty much any other molecule the plant needs. It can be turned into, it can be combined with fructose to create sucrose. Which is what the plants use to transport carbs from one part of the plant to the other. It's also the starting point for the synthesis of starch and cellulose. Starch being that long-term storage that we talk about in roots that we saw in the potato. And it can also be a hydrocarbon skeleton to form fatty acids that we see in plant oils like corn oil and sunflower oil and olive oil. So that G3P can be used for a lot of things. So that's why the Calvin cycle is so important, because it generates that G3P. So there are other types of photosynthesis. There is something that we call C4 photosynthesis. So C3 photosynthesis is things that are going to be like wheat, rice, oats, and things that have the ability to take in that CO2. But in hot, dry climates, those stomata have to close where they take in CO2. And that decreases your CO2 and increases your O2. And it leads to like a less efficient version of photosynthesis that we call photorespiration. So to solve that problem, some plants have adapted, and those plants would be C4 plants like sugarcane and corn. And so they fix CO2 to a C3 molecule and wait until a later time to be able to process that when they can get some more CO2. So you can see here the difference between a C3 and a C4. You see how these are where they can get a lot more of that solar energy, whereas these are kind of inside, and so it's harder, especially for these right here, to be able to get that solar energy and to be able to absorb that CO2, especially when they have to close those off. And so they have to come up with a different photosynthesis to be able to produce what they need. So where CO2 fixation in like C3 plants happens with that RUBP, in the C4 plants, they have to store that CO2 before they can move it into the Calvin cycle later. There's also something called CAM photosynthesis, which is crassulacean acid metabolism. And they are going to be things like cactus. So during the nighttime when it's cooler, they fix that CO2. So they can open those stomata and they can bring in CO2. And then they store the 4 carbon molecules in large vacuoles. And then during the daytime, they use the NADPH and the ATP that they get from the sun to be able to put that CO2 back into the Calvin cycle so that they can create their G3Ps, which are what creates the cellulose and things that they need. Each method of photosynthesis has advantages and disadvantages, and a lot of times they depend on the climate. C4 plants are most adapted to high light intensity, so places where there's going to be a lot of light, high temperatures, and limited rainfall. So like grasses are a good example of C4 plants. C3 plants are better adapted to the cold and high moisture. So like we were talking about those things like rice and wheat and oats. And then CAM plants are better adapted to extreme aridity, which is the dryness of the desert. And that's going to be things that are like cactuses and succulents. So we've talked about photosynthesis. Now let's talk about what happens in an animal cell, which is cellular respiration. Not to say that cellular respiration doesn't occur in plant cells because they also have mitochondria, but it happens to a lesser degree than an animal cell. So here's the connection between photosynthesis and respiration. So in photosynthesis, we're taking in that light energy and we're producing oxygen molecules, remember? Well, that oxygen molecule and those organic molecules like glucose and starch are what is needed for cellular respiration in those mitochondria. And then they give off heat energy. So there's two types of cellular respiration. There's fermentation and then there's cellular respiration. Anaerobic means without oxygen and aerobic means with oxygen. So cellular respiration oxidizes sugar and produces ATP in 3 to 4 main stages. Glycolysis, which happens outside of the mitochondria, and then the prep stage or the transition stage, which happens inside the mitochondria. We've gotten away from saying Krebs cycle, and we now call it the citric acid cycle, but Krebs is who discovered the citric acid cycle. And then the ETC, which stands for electron transport chain. Fermentation, on the other hand, oxidizes sugar and does not produce ATP, but it yields a constant supply of that NAD+, which, if you remember from photosynthesis, helps to power that electron transport chain. So you can see here, this is a breakdown of basically what happens in glucose breakdown or in cellular respiration. So electrons are removed from substrates and received by oxygen, which combines with hydrogen to become water. And glucose is oxidized and oxygen is reduced. So you start out with glucose and oxygen and you give off CO2 and water. So what are NAD and FAD? NAD plus and FAD. So those are some enzymes and coenzymes that are necessary for cellular respiration to go faster, basically. So NAD plus is a coenzyme that carries electrons, and it helps the enzyme do its job and participates in the reaction. The NAD plus receives 2 electrons as the substrate glucose is oxidized. So if you remember, the electron is received by NAD as part of the hydrogen atom, and the hydrogen atom consists of a hydrogen ion and an electron. So NAD becomes NADH. And they use them over and over again. In contrast, FAD, while it is another coenzyme, and it's frequently used as an electron carrier, it doesn't produce as much ATP as an NAD does. So ATP is known as adenosine triphosphate. So the T stands for tri, as in tricycle, like 3, and adenosine diphosphate, which is when you get what you have after that phosphate breaks off. Di stands for 2, so it just has 2 phosphates attached to that adenosine. And that bond you can see is a high energy bond. So what that means is it releases energy when it's broken, and that's what your muscle cells and your heart cells and your liver cells use for energy, is that energy that's released when that bond is broken. So cellular respiration includes 4 phases. Glycolysis is where we break down that glucose into 2 molecules of pyruvate, because your mitochondria can't actually use glucose in its original form. So it has to break it down from a 6-carbon molecule to a 3-carbon molecule, which is what pyruvate is. That occurs in the cytoplasm, and ATP is formed, but only 2 ATP is formed because it also requires ATP to break it down. It doesn't require oxygen to do glycolysis, so that is another form of anaerobic respiration. The next phase is called the transition phase or the transition reaction. And both of those pyruvates are oxidized so that they can enter the mitochondria. And so when this happens, it forms a 2-pyruvate are added to something called coenzyme A, and that makes them into 2-acetyl coenzyme A and 2 carbon dioxides. And electron energy is created because it's stored into that NAD plus that we talked about, and 2 carbons are released as a carbon dioxide, 1 from each pyruvate. So then we enter what's called the citric acid cycle. So we take those 2 acetyl-CoAs, and that does happen inside of the matrix of the mitochondria, and it produces NADH and FADH2. It's a series of reaction that also releases 4 carbons as CO2, and it turns twice, once for each pyruvate. It produces 2 immediate ATP molecules per glucose molecule. So you can see between glycolysis and the citric acid cycle, we're not getting a ton of ATP. We're only getting 4 total. So where are we going to get the rest of our ATP? Well, that's going to come from the electron transport chain. So every time that an oxidation reaction happens, once we input that NADH and that FAD, it's going to release a bit of energy as it runs down that chain to change that ADP into ATP by chemiosmosis, the same process that we saw in photosynthesis, where it's using that hydrogen concentration gradient to push that ATP synthase. And it produces 32 molecules of ATP. So you can see that glucose goes to pyruvate here, and it enters into the mitochondria, and then it is used in the citric acid cycle and the electron transport chain, and eventually either 32 or 34 ATP are produced from that electron transport chain. It changes based on what type of cell it is, like whether it's a muscle cell or a liver cell, how many you actually produce. So let's break down each of these steps a little further. First, you have glycolysis, and it occurs, like we said, outside of the mitochondria. So you have to use 2 ATP to activate the glycolysis step. So we're having to burn some energy in order to create energy. So kind of that whole, you can't make an omelet without breaking some eggs kind of idea. So glucose splits into 2 G3P molecules, which is that pyruvate, after the ATP gets that activation energy going. And then when that G3P is oxidized, because we remove some electrons and hydrogen ions, those go into that NADH, which we can use in the electron transport chain later on to make some more ATP. So 4 ATP are produced by phosphorylation, and we have a net gain of just 2 ATP because remember we had to use 2 to get started. So you can see here the electron transport chain. And here we see some basics of glycolysis. You're putting in the glucose, you get out 2 pyruvic acids. So here's a input-output chart of the glycolysis. That would be probably pretty important for you to write down. And if you need to pause the video here, that'd be a great time to do so. So pyruvate. Pyruvate is a pivotal metabolite in cellular respiration. If oxygen's not available to the cell, then fermentation occurs. And this is where it uses pyruvate to create some more ATP, but not as many as you can get through the aerobic cellular respiration. It's incompletely metabolized into lactate. So like when you're running, your oxygen isn't sufficient to drive these reactions. And so it goes into this anaerobic fermentation and lactic acid builds up. And that's what causes that burn in your legs. Other forms of fermentation would be like alcohol fermentation, where yeasts produce carbon dioxide and ethyl alcohol. We see this in bread when we use yeast to make it rise, or we see it in when we make actual alcohol. But you have to be careful and continue to add yeast to your wine or beer, because if you don't, then that alcohol eventually kills it off the yeast itself. Lactic acid fermentation is also carried out by certain bacteria and fungi to produce, like we said, that lactic acid. It's commercially used in cheese, yogurt, and sauerkraut. And there's some other chemicals that bacteria produce when they perform this fermentation process that we actually use, like isopropanol, which would be like your isopropyl alcohol that you use, butyric acid, propionic acid, and acetic acid. Acetic acid is vinegar. So it's an alternative to aerobic, and you can see that when it breaks it down, you don't get as many ATPs. But your cell has to have a way of replenishing that NAD, or else it won't continue this anaerobic respiration. So in lactic acid fermentation, which is what our bodies do when there's no oxygen, pyruvic acid is converted to lactic acid, and the NAD is recycled. Lactic acid fermentation is also used to make cheese and yogurt, two of my favorite dairies. So here's another one of those input-outputs of fermentation. Pyruvic acid is chemically transitioned into another molecule, that acetyl-CoA, to be put into the Krebs or citric acid cycle. And so it's broken down and it forms CO2 and then that acetyl group, which attaches to that coenzyme A. So then you have acetyl coenzyme A. And when it goes through the citric acid cycle, remember, it doesn't produce that many ATP, but it produces enough. And then it completes the oxidation of that glucose that generates a bunch of those NADH and FADH2 molecules that we're going to use in that electron transport chain. So we're going to use the products of the citric acid cycle to make even more ATP by using the hydrogens off of those NADHs and those FADH2s to create that hydrogen gradient concentration. When those hydrogens diffuse back through the inner membrane through that ATP synthase that we talked about in also in photosynthesis, it captures that energy to make that ATP. So here is an overview of cellular respiration. This is probably the best way to kind of visualize it. So you take in this 6-carbon glucose and it undergoes glycolysis in the cytoplasm. Remember, that's where it splits it into those 2 3-carbon pyruvates. And you're going to generate 2 net ATP because you got to insert some ATP. But it also creates those NADHs that we're going to need to use in the electron transport chain. And then those pyruvates go through the Krebs cycle after they're prepared into that acetyl CoA, and you're going to produce some CO2, but you're also going to produce ATP just from the Krebs cycle, and even more NADH and FADH2. And then it's going to go through that electron transport chain and make 32 ATP. So your net harvest is going to be 36. Now, cells use lots of different organic molecules as fuel. We often think of carbohydrates, but you can also use fats and proteins. That's how the keto diet works, is it instead of breaking down glucose because you're not taking it in, it starts to break down proteins. And those proteins are digested into amino acids, which are chemically altered and then used in the Krebs cycle. And then fats are broken up and fed into glycolysis that can be used into the Krebs cycle as well. So what you need to remember as well is that the fuel for respiration, cellular respiration, ultimately comes from photosynthesis. If we didn't have those photosynthetic plants creating those carbohydrates, we wouldn't have any of those carbohydrates to take in to be able to power that cellular respiration. Remember that plants are the only ones that can make their own food, and we have to eat what they make us, basically. So I hope that this has been a helpful overview of both photosynthesis and cellular respiration. Don't forget to do your homework. And if you have questions, please bring them to class so that we can talk about them. Because I promise whatever question you have, your buddy also has. So have a great rest of your weekend.